Hello, everyone. I'm Virginie Goubert, Global Application Specialist, and I will present you today how to move your Opal assay from manual to automated staining. So, as you know, Akoya Biosciences has been the pioneer in spatial phenotyping. What do we mean by spatial phenotyping? We really saw, the gap between genomics and proteomics technique, where you get a lot of information about gene expression and different protein markers, but most of the time you have an approximation of the location of the cells, or you completely lose the tissue context. Whereas in immunohistochemistry, you're only looking at one marker at a time, maybe two. You do have the tissue context, but you're missing some phenotyping information. And so we really further developed this concept of spatial phenotyping for enabling researchers to look at several phenotypes within the tumor microenvironment at a single cell resolution. And for that, we have developed two platforms, Codex and Phenoptics. So Codex is our ultra-high plexing multiplexing tool. So it's a discovery tool that you can plug to any inverted microscope, and it's fully automated. And it will give you the ability to look at 44 markers at a single cell resolution. But when you need to sample, to look at several samples in one day, to a large cohort or high volume of samples, you need a higher throughput. And that's where Phenoptics come into place with Opal multiplexing and Vectra Polaris. And that's what I'm going to focus my talk about today. So just to give you some background about the Vectra Polaris before I dig into the technical part of Opal staining, Vectra Polaris has been really designed from the ground up to really respond to clinical needs and requirements. We're now super excited to have the certification ISO 13485, which is done for medical device manufacturing. It has a throughput and reliability one need when it comes to high volume clinical research projects with this fast scanning and multiplexing capability. So in case you don't know yet, but Phenoptics workflow, it really comprises different pieces of puzzles, from staining reagents with multiplexing reagents that we call Opal, that you can fully automate, and that's what I'm going to present today. The imaging part with Vectra Polaris for a very fast multispectral imaging modality. And we also have a full data analysis software with different solutions, and our newly developed Proxima, which is cloud-based for data analysis, a fast data analysis on the cloud. We're also open to third-party software. If you want to work with Hello or Visiopharm, it's also possible. So what is Opal? I think, if you're listening to this webinar and have some interest going from manual to automation, you might have some background, but Opal is based on TSA. So it's pyramid signal amplification, which means you do have an amplification. And on top, you can multiplex by a repetition of staining cycles. Every staining cycle is comprising the same steps. So it's always a blocking step, then a primary antibody incubation step for 30 minutes, then washes, then secondary for 10 minutes, then washes and Opal dyes for 10 minutes. And then we strip the antibody complex. Only the Opal dye, which is covalently bound, will remain on the targeted epitope. And then you can target another epitope with another set of antibody. And the good thing out of this repetition and always coming fresh is that you can use many different primary antibodies from the same species. So if you want to use only rabbit-raised antibodies, you can, and that's possible only with Opal multiplexing. So now let's go into the automation part. So as you might know, we have validated our Opal protocol on the Leica Bond. And we also have many users worldwide successfully using the Roche Discovery Ultra, going to 6-plex or even 8-plex using the Opal dyes on those autostainers. And of course, when you automate, you speed up your research, you speed up your assay because you can run it overnight in 12 hours instead of 2 or even 3 days when you do an 8-plex manually. You improve your reliability and your reproducibility because it really decreases the CV compared to manual assay. And when you bring some technician variability, it could also be an issue moving into clinical trial, for example. And it also brings you flexibility because you're really flexible in the antibody you use and the dyes and so on. The protocol is, as I said, a repetition of staining cycle. The only difference now with our new Opal 780, our new dye to be imaged on the Vectra Polaris, which is Opal 780, we have to always place this dye at the end. So that's why you will always see, whatever the marker you're pairing it with, you will always see this TSA dig and Opal 780 detection placed at the end as last in the sequence. And the reason is that the Opal 780 is an antibody, is anti-dig, which means we cannot perform any stripping afterward. If not, you would remove it and you will lose the signal. So that's why it has to be placed right before DAPI at the end. So we offer a full spectrum, I would say, of kits, so of detection kits. So we have a 4-color kit, manual and automation, 7-color, also made for automation. And you can go up to 9 dyes by buying 2 more dyes if you want to do 8-plex plus DAPI. We also have a kit dedicated to mouse tissue with an anti-rabbit secondary conjugated to HRP. But also, I want to take this opportunity to give to let you know about our new panel kits. So those kits are ready to use. They've been fully optimized on the Leica Bond. So it comes with a ready-to-use Leica Bond protocol. And it's to be used and imaged on the Vectra Polaris. Sorry, jump to the next one. But if you want to go up to 8-plex, you can also add, as I said, 2 more days. For example, here, on top of our panel kit, you can think about Ki-67 to look at proliferation of cells, or add a B cells marker with CD20. And then you have already a lot of data out of one tissue section. So Opal really brings you a maximum of flexibility, not only from the antibodies you use, so you can easily use your favorite antibodies, the ones that you have pre-validated in your labs and that you trust, or you can use ready-to-use pre-optimized panel kits. Those are the ones that I just showed you. And you can do the assay manually or on any, I won't say any autostainer, because the autostainer has to be quite flexible. And so that's why we're limited to the Leica Bond RX or the Roche Discovery Ultra, because they're quite flexible autostainer. I just also want to give you a quick introduction to RNAscope that you can also implement onto the Leica Bond. And since I have more and more requests about how to perform Opal assay with RNAscope, I just wanted to let you know that's also possible to automate FISH, so RNAscope detection, plus an antibody. So Opal protein detection, and all combined and automated on the Leica Bond and Roche Discovery Ultra. So if you want more info about this protocol, please reach out to your Biotechny, ACD FAS, field application specialist, or to Akoya field application specialist. Just to give you a flair of what we could have in terms of RNA detection with the Vectra Polaris. So that's kind of a blend image that you will have without autofluorescence isolation. And that's what you get when you apply the Opal multiplexing, Opal amplification, and autofluorescent subtraction with the Vectra Polaris. And that's just a representation you can get as a what we call pathology view with Inform. So now the goal of this webinar again is to teach you how to go from manual to automated staining. So let me go straight into it now. So first, let's not forget that, although Opal detection can sometimes do miracle in terms of amplifying low expressors or really giving an extra boost to your staining, it's really important to treat your tissue block well before moving into the staining parts. So that's why it's really important to think about the tissue fixation, storage, and epitope retrieval. We also have some recommendation in terms of slide preparation. So the dewaxing is sometimes not enough in the Leica Bond or Roche Discovery Ultra. And so, for example, internally, we like to still bake the slides overnight at 55, 60 degrees in the oven. You can also do it 2 hours before performing the staining. And it's just a way to melt most of the paraffin away before performing the dewaxing into the machine. It's also really important to use good slides. So I really encourage you to use a Superfrost Plus or Tomo with pre-coating to make sure the tissue really holds onto the glass slide. Also, as I said, the epitope retrieval is also important. So it's really important to pre-validate if it's better to use pH 6 or pH 9. Also, in our kit, we have some buffers included, so blocking solution, antibody diluent, but please feel free to use also or to test antibody diluent or blocking solution of your choice. If that's something you validated manually or using DAB, there is a big chance that it works quite well or that it's also needed when you go into the autostainer. And either autostainer like our Roche will give you this flexibility to add or not an extra blocking step or to add your own blocking solution to perform hydrogen peroxide if your tissue has endogenous peroxidase in it. So feel free to experiment if you have too much background. So now comes the choice of the antibody. So when you move from manual to the autostainer, you probably already have an idea of what concentration to use for your antibodies. But since it's still not exactly the same, you have some dilution factor. For example, in the Roche, you have 1-to-3 dilution factor. And often I've seen that you have to readjust a bit the concentration of your antibodies when you move into the Bond RX. So I would encourage that you do a small titration. So from the manual optimal dilutions you found, you can do two times more, 2 times less, 4 times more, 4 times less, so that you see what gives you the best pattern and the best signal to background. In order to do this titration, you can use the Opal dye at 1 to 150 on the Leica Bond RX or 1 to 50 on the Discovery Ultra. And the reason we do 1 to 50 again is because we have a dilution factor happening in the Discovery Ultra, which means that we have to increase the concentration of the reagents a bit. At this stage, again, do not worry too much about the balancing your signal or looking at the signal intensity. What counts is really to look at the best signal to background and which one will give you the best signal to noise ratio and pattern. So looking at the image, you can hover, if you're using Inform, you can hover onto the tissue and see those specific ones from the background and look at the specific signal and divide it by the background. And that will give you your signal to noise ratio. And we always intend to have more than 10 SNN signal to noise ratio of minimum 10 for Opal. Also with Inform, you can look at the pattern in a DAB mode. So the fluorescent signal is represented as DAB. And so this way you can really compare what you obtain with DAB or what you obtain manually. Also, if you need to perform any stripping, stripping efficiency experiment, let's say it's the first time you experimented on the Leica Bond or the Roche Discovery Ultra, and you're not sure if the temperature is high enough or if the stripping is complete, you can do this easy test, which is to complete a whole staining sequence, do the denaturation, so you perform the stripping, and then you come with a second sequence with a different dye and without any primary antibody. Since you don't have a primary antibody in the second sequence, you shouldn't see any of the second dye colocalized on the first one. So that's the easiest way to do. If not, you can just also start with your experiment. If you don't have any markers which are supposed to colocalize, and you do see a colocalization, most of the time it's a stripping issue. If it's not an unmixing, spectral unmixing issue. And if that's the case, if, for example, you do see an issue with incomplete stripping, you can increase the temperature or increase the incubation time. Talking about stripping and repeated heat treatment, so we have also some recommendations in terms of how to define your order of addition. So if you have done it manually, if you already have a pre-optimized order of addition and you have tested it manually, whatever the heating method or the stripping method, you have a big chance that the order of addition would be also optimal this way in the autostainer. But in case you haven't done it before and in case you're not sure, you can perform again this what I call a heat epitope retrieval experiment. So you look at the effect of those repeated heat treatments on your targeted epitopes. And the best way I found to do it is to have 3 serial sections, one section that you will stain after 1 epitope retrieval, one that you will stain after 3 heat or epitope retrieval, and one after 6. And you look at the signal, and depending on the signal and the signal to noise ratio you get, you can define if actually your epitope benefits from having several retrievals or is damaged, and in that case, you will have to place it first. From my experience, it's only 10% or 10% of epitopes which get damaged through the heat treatments, and so it's really rare that you always have to place it first in the sequence. So again, you look at the staining pattern, you look at the signal intensity, you calculate your signal to noise, and then you see where to place your epitope in the sequence. And most of the time, what users do is to build up an Excel file where you can have this experiment registered. And so you can say, okay, for example, CD20 is good, whatever the position. It can be added first, third, or sixth in the sequence. It's always good. But some, for example, HLA-DR, it's better when it's placed first, and the signal is not so good when it's performed after 6 epitope retrieval. And in that case, you know, it's better on the first. What you don't have to worry in the autostainer, and this experiment was done on the Leica Bond, is about the opal dye stability. Whatever the opal dyes, they're very stable in the Leica Bond or Roche Discovery Ultra. So you don't have to worry about placing, I don't know, 520 first or last in the sequence. Just keep in mind that Opal 780 is the exception. It's the only one which has to be placed last in the sequence, just because it's an antibody. Okay, now last kind of part is the detection part. Like, how do I pair my Opal dyes to my markers? And we have some rules for that. That's the kits we have. So that's the kits that are pre-made for automation. So we have a 4-color kit and 7-color kit. So this way you don't have to worry about which dyes to use. We provide already the optimal combination of dyes, and we provide everything you need in terms of buffers, solutions, diluents to perform your assay. The only thing you will have to use is your primary antibodies, of course, if you want to use your own primary antibodies, or you will have to use the ER1, ER2, so epitope retrieval solutions from the autostainer vendor. So, or two, I would say, rules, condition when it comes to choosing which dyes is to first know if your markers will colocalize. If you're targeting colocalized markers, I recommend that you use spectrally distant opal dyes. So I give you an example. If you have CD3, CD8, use 520 and 620. Don't use 520 and 540 if possible. And also, you have to look at the abundance of markers. I mean, the abundance and expression, overall expression of your target. So, for example, cytokeratin is quite well expressed, quite abundant, so it can be paired to a dimmer fluorochrome. But the low expressors really have to be paired as much as possible to a bright fluorochrome, fluorophore. And you have a table that we're always happy to share to give you the level of brightness of our different opal dyes in our multispectral instruments. So Mantra, Vectra Polaris, or Vectra 3, because you have slight difference between the imaging instruments. So we're really happy to share those with you. Again, as I said, it's doable to look at colocalized markers with the autostainer and with Opal. Just make sure you keep your amplification and deposition quite low. Don't oversaturate your epitope. And again, make sure you don't use spectrally adjacent dyes. Use as much distance as possible between the dyes to make sure you can really unmix reliably your dyes. So for example, here CD45 row, CD8, CD3, CD45 LCA, they're all on the same cells. And what you can do is have 520, 570, 690, and 780. And this way, you're fine in terms of spectral distance. So last, once you have done And this move from manual into the autostainer, you know exactly now which dilution to use for your primary. You revalidated your Opal, your order of addition. You know which dyes to use depending on the intensity you get for each marker. What you have to make sure is that it's all quite well balanced because we have the tendency to have an amplification with Opal, and so sometimes we really have too high signal. And in that case, you just have to dilute your dye some more to make sure it doesn't go above 50 counts. So 50 counts is really the maximum for Opal 480 and Opal 520. But for the other dyes, I would even encourage that you stay below 20 counts to avoid any unmixing artifacts. And for Opal 780, since near infrared, it's very dim, don't expect to go higher than 10. You're happy if it's below 10, and usually the signal to noise ratio is very good for this dye. I just want to finish now this presentation on some recommendations about maintenance of the system. Since we have more experience internally on the Leica Bond, I will restrict those recommendations to the Leica Bond. I'm sure you know it's also very important to well maintain the Roche Discovery Ultra to clean inside, and I will let specialists talk about this part. But internally, we have seen a huge impact on cleaning and maintaining the system as much as possible to have reliable data and good CV, good coefficient of variation. So after each run, we remove the reagent tray, we clean outside the probe, and we clean everything, wipe off everything, the heating plates and so on with 70% ethanol. We also do a clean fluidic step after each run. We clean or even change cover tiles, especially if you have precious samples after each run. And we discard all titration inserts. But I think that's also the recommendation from Leica. We also clean the aspirating probe on a weekly basis using the clean aspirating probe process incorporated into the interface. And we, of course, have to use the aspirating probe cleaning kit from Leica. We also see internally that it's better to shut down on a regular basis the Bond RX and the computer. So we usually do it over the weekend. And from time to time, on a monthly basis, it's good to clean the bulk reagent containers as it's also outlined in the Bond RX manual. And if you really see a CV going above 10%, which is because we do have really good CV with the Leica, with our Leica Bond RX internally, but if you do see a lot of variability, ask the service engineer to come and change or replace your syringes and probes. Also, what could happen due to the way it's dispensing on the slide is a gradient issue. And if you do see a gradient issue, we also have some guidance. So first, double dispense Opal reagents. So we do a 0-minute flush followed by a 10-minute incubation. You can also, in that case, further dilute the dye. So instead of using 1 to 150, you can go up to 1 to 200 or increase a bit more. If you do double dispensing, that would save you a bit of reagents. And if really that doesn't solve the gradient issue, the gradient signal gradient issue, then you can do double dispensing on all reagents, including antibodies. If possible, try to put the tissue section on the first top, medium top of the slide instead of in the bottom. And for DAPI, we also recommend that you do open dispense and also double dispense. So open dispense is available if you have the version 6.0, you can easily change. If not, you will have to ask the Leica engineer to set it up for you. But yeah, it really depends on the tissue section, the thickness, the topographical variability. You know, it's quite depending on the tissue block you have. We also saw that increasing to 97 degrees for stripping is also bringing a better stripping efficiency and could also improve the signal gradient issue. So with that, I will conclude my talk, and I encourage you also go on the website. It's very well made with a lot of articles, a lot of testimonials, some information about our automation kit, and some protocols as well. And I'm happy to answer any questions. Thank you. Thank you very much. I hope you found my presentation useful, and I will now open the ground to questions. So as you can see on the screen, please use the Q&A functions at the bottom of the screen to type your questions. So I see a few questions already, so I will start with the first one. On what basis the order of antibodies are chosen for staining? So that's a really good question. So indeed, all antibodies are not performing the same after several antigen retrieval. So it's a really good or important thing to first test how the antibodies are going to react. And so the best way I found to do it is to have 3 serial sections, one that I treat after 1 heat treatment, so 1 epitope retrieval, one that I treat after 3, and 1 after 6. And I see if the signal increase, if I have a better pattern. Then if that's the case, I know I can place it at the end of the sequence. If I see a drop in signal or if I see an increase in background, then I know this one has to be placed first. So I hope it answers your question. Can I use mounting media with DAPI for Opal protocol? So if you're not using a multispectral imaging system, yes, you can. That indeed will save you a step at the end. But if you're using a Vectra Polaris or a Vectra, any multispectral imaging system where you're doing some spectral unmixing, I will highly recommend you don't use DAPI included in the mounting medium, because the system will detect some DAPI left over everywhere on every pixel. So you will have a bluish veil on your slide. So you want to avoid that. So a question from Anaïs. So hi Anaïs, thank you Virginie. What are the recommendations for the cover slip mounting? So in R&D and in our service lab, we use the prolonged diamond from Thermo Fisher. You can also use prolonged gold, but I think the diamond gives better results, especially for the near infrared dyes. So yeah, prolonged diamond is a good one. Can I use DAB to test stripping instead of the Opal dyes? Indeed, you can do that. The only thing is, if you're not using the same secondary as you're using for DAB, you wouldn't know if the whole antibody complex, primary and secondary, is gone when you're in your Opal situation. But indeed, if you want to just test for the first time while saving some Opal reagents, you can use DAB to test stripping. That's indeed a good way. When testing antibody signal at different numbers of antigen retrievals, would it help to use DAB as a detection? also, if I'm using just only one fluorophore for testing, can safely change fluorophore after it is optimized. Yeah, so you can also see the pattern changing with DAB, but really Opal will give you the dynamic range to really appreciate if you have an increase in signal after several antigen retrieval or a decrease. So for this antibody signal, antibody resistance to epitope retrieval, I will encourage you to do it with Opal. I mean, it's not really antibody resistant. It's more the epitope that the antibody recognize, which might be affected. And you can indeed do that with the same fluorochrome. So if you have, I don't know, some leftover of Opal 520, or if you know for that Opal 520 gives you really good signal on your microscope, you can indeed choose this one to perform all your first optimization steps. And if you change it later on and you see a change in signal, as I said at the end of the presentation, you can always adjust, rebalance the signal by adjusting the Opal dye concentration, or the Opal dye dilution. Can I mix between ER1 and ER2 for Opal? Indeed, that's also a good question. Yes, you can. Just be aware that ER2, which is pH 9, is a bit harsher retrieval, which means some antibody might show a bit more background after ER2. So just keep in mind that, you know, everything that would be behind ER2 will be ER2 retrieved. So if you have an antibody which really needs pH 9 for epitope retrieval, I will try to place it at the end so it doesn't affect any other epitopes before then. And also, yeah, so I also, from my experience on the field, I also noticed that often antibodies which require pH 9, so ER2, they're really good placed after several ER1, so pH 6 treatment. So I think a good pH 9 is equivalent to several pH 6, if that makes sense. Another one, are the protocols for Bond on your website updated? Our Bonds run the latest software version, but the protocol instruction on your website did not match. So indeed, we have a lot of resources on our website about how to set up the protocol and so on. But the version 6.0 have the previous version, so with the previous Opal dyes. But please contact your local specialist. We can share with you some protocols and exchange also with the Leica FAS specialist to make sure we get you up and running in a fast manner by sharing the latest version of the protocol. If I don't have access to the Inform software for unmixing, are there methods you recommend to reduce autofluorescence but still retain the signal intensity? Indeed, so having multispectral imaging and using Inform really enabled to completely isolate the autofluorescence from the tissue, the autofluorescence you get from the tissue, from the specific signals you get with Opal. But if you don't, I heard you have several techniques like photo bleaching or what was the other one? Something black. Yeah, I can come back to you on this one, Alicia. But yeah, you have, I know, Sudan black. You have several methods now to reduce, but it's never completely the same, of course, as using Inform, but that's a good way around it. Can I replace TBS, so trace buffer, with bond wash buffer for Opal protocol. Yes, so you can also replace the twist-based buffer with the bond. It's, I think, pretty similar, but I will contact Leica to make sure that's okay to use that. But for Opal, there is no difference between the bond wash buffer and what we see with the handmade TBS buffer. Did you test the, so Melanie, did you test the RNAscope with the Opal kit on Discovery Ultra? I haven't personally, but I know some people have done some RNAscope and Opal on the Roche. It's not as published and seen as on the Leica, but I think it's doable. But unfortunately, I don't have a protocol or anything, but I would be happy to get in touch with you, or you know, I'm sure if you contact Biotechny, SCD specialists, they can also get in touch with us so that we can work something together. Ventana, so another one from Mariam. Ventana Discovery Ultra multiplex protocols usually start with CC1, and then there is only choice for CC2 for stripping steps. Could you comment on that? Yeah, so indeed, I mean, it depends on the versions you have, but the Discovery Ultra is usually quite flexible now on the newest version, where you can choose between CC1 and CC2. For antigen retrieval and for stripping steps, most of the time, most of the Roche Discovery Ultra users are really just staying on CC2 because it's just 8 minutes for 100 degrees, and it's the most efficient stripping technique Roche has found to remove the antibody. So another question from Mariam. Would you have some example protocols for Discovery Ultra? So yes, indeed, we do, and I would be also happy to share. We have some published data, and we have also some protocols that some users were nice enough to share with us. So last question. Hi, if I'm using my buffer to antigen retrieval, do you recommend adding H2O2 step for background subtraction of HRP? Indeed, that's a very good point. H2O2 is blocking endogenous HRP present in the tissue. So whatever the method, you know, manual, automated, Leica or Roche, there is no harm in adding a H2O2 step in your protocol. If you, for example, see background for maybe those endogenous HRP, since Opal is based on HRP, indeed, it would be useful to add such a step. Yeah, another question, can I use different secondary antibodies and not only the one provided in the kit? So indeed, we try to provide all the buffers and reagents you need to perform your assay in our Opal detection kit. So we provide this mixture of anti-mouse, anti-rabbit, HRP conjugated secondary. But if you're using a goat, for example, or if you have a well-performing secondary working with your primary, please feel free to use it or to test it in parallel to our secondary. Because as long as the secondary is conjugated to HRP and preferably polymer HRP, that would give you even a higher amplification because you have several HRP molecules instead of one. So if you can choose a polymer HRP, yes, you can use any secondary you'd like with our Opal dyes. So I have another question now about the Discovery Ultra. If you are applying the Opal reagents manually on the Discovery, do you still have to consider the dilution factor? Or again, an excellent question. because indeed on the Roche Discovery Ultra, for what I know, there is a 1-to-3 dilution due to the addition of the reaction buffer at every step. So to inject the reagent under the oily cover tile, they're using reaction buffer. And this reaction buffer is indeed diluting the reagents a bit. So we think it's about a 1-to-3 dilution. So indeed, you have to take this into consideration and increase a bit more your primary antibodies or eventually your Opal dyes. So let me see. I also see a question in the chat function. So how does it work for TMA slides? Since they're thinner and might have more, might give more background. Opal work on any type of tissues. I mean, now it's really been around for, I don't know, many years. Lots of publications on diverse tissue sections, organs, tumor, healthy, bone, marrow, brain, whatever the tissue is. And we haven't seen any issues. So TMA course, I mean, a lot of people are doing Opal on TMA. TMA is usually actually a bunch of different tissue types, different tissue blocks, and we haven't seen any issue. And if you do have background, we also have way around it. As I said, different blocking solution, or if the background comes from HRP, you can use this H2O2, so hydrogen peroxide solution, as I said earlier for a question. And yeah, you can you can find a way to decrease background. And if you're using multispectral imaging, you can also decrease background by subtracting the autofluorescence out of the most fluorescent core, for example. Let me check. So there's nothing in the chat, nothing more in the Q&A. Yes, so I think we're reaching the end of this webinar. And if you have any more questions coming up, please write us. So you can directly write to me or contact your local specialist. We will be really happy to answer your questions. And with that, I wish you a good day or a good evening. Thank you very much.